Kind words of introduction and for the invitation to come here and tell you a little bit about, well, Max asked me to talk about cryobiology as an emerging longevity technology, and it's a great pleasure to finally be in person at ARDD. I've given a couple of talks at ARDD online, but this is my first one in person. And so, as I said, I was asked to give a talk on cryobiology and cryopreservation. As I'm sure you're aware, we can cryopreserve biological materials to some degree. I mean, many of us are biologists. I'm a biologist by training. We can cryopreserve cells, for example. So the problem, however, is that when you lower temperature below freezing point, you'll have ice formation. So you have these ice crystals that form that can burst cells. They can damage all kinds of cellular components. And that is one of the major problems we have in cryobiology, in the cryopreservation of biological materials. Having said that, there has been some successes. It's not working, but there has been some successes. For example, we can cryopreserve embryos. So there has been cases, this is, last time I checked the record, was what embryos frozen 30 years ago bring back to life. So when someone tells you that nobody has ever been brought to life from being frozen, that's not entirely true. People have been frozen before for 30 years and then brought back to life, with a caveat that happened when they were very little, tiny, 100 cell or so embryos. But that has been done. Now, the problem, of course, with cryobiology is when you start to apply these technologies to larger structures, it's essentially a problem of physics. It becomes a problem because it takes longer for the temperature to drop. Now, in cryobiology, we use cryoprotectants to prevent ice formation. But if it takes very long for the temperature to drop, then you have to use higher concentrations of cryoprotectants, and it becomes toxic. So it has a longer exposure, higher temperatures, longer exposures, higher concentration of cryoprotectants, it then becomes toxic. So, I mean, we published this big review a few years back in BMC Biology on different problems and challenges, but also prospects of cryopreservation that summarizes the current state of the art, which is that we can cryopreserve cells and small tissues and embryos and corneas. So tiny structures, biological structures, we can cryopreserve them. As I said, with cryoprotectants, but there's this major issue, this major bottleneck of scaling up to larger organs. Again, because it's physics. If you have a larger organ, it takes longer for the temperature to lower, to cool down. And the problems of cryopreservation are well established. We need to prevent ice formation, which as I say, can be done to some degree with cryoprotectants, mitigating cryoprotectant toxicity. So, as I said, cryoprotectants like DMSO, ethylene glycol, and so on, they're relatively safe. They don't have very high toxicity, but they have some toxicity. So at the very high concentrations and for long exposures, they become toxic, and that's a major problem. And it's a major problem in cryopreserving organs. So when you talk about large organ preservations, that's unfortunately not possible yet. We cannot cryopreserve large human organs yet. And this is a major problem. And I don't think people are aware of how big of a problem it is. So the situation happens now is that the vast majority of organs for transplantation are lost. So, you know, as I'm sure you're aware, there's many patients that require organs, liver, kidney, hearts, lungs, and so on. But the vast majority of these are not used. So, to give an example, hearts, only 30% of transplantable hearts are used, and only 20% of transplantable lungs are used. That means that the majority of hearts and lungs that are available for transplantation are actually lost. And the reason is that, well, if someone dies in an accident, for example, and you have organ transplantation, they're lost because you cannot preserve them long enough until you find a suitable donor. So that is a major bottleneck, and it leads to many lost lives. So, in fact, how many lost lives? That's something that Sebastian Giwa et al. They estimated in this big review in Nature Biology a few years ago. So they estimated. This is data from the US, but they estimated that there's deaths from end-stage organ disease is over 700,000 in the US alone. So what it means is that if we could prevent those, if we could have organ replacement that could prevent those, it could prevent over 30% of all deaths in the US. So in effect, it could double the likelihood of someone living to the age 80. So it would have a massive impact. Now, what that means in practice is if we could safely and effectively cryopreserve organs, then what it means is that essentially if someone dies in an accident, for example, you would cryopreserve their organ. You wouldn't need to find a donor, sorry, find a recipient right away. You could cryopreserve their cryopreserve their organs, and then you'd have all the time you would need to find a suitable patient, a suitable recipient. And that would make the process much more efficient, and you wouldn't lose all this 80 or 70% of lungs and hearts that are lost because we can't safely cryopreserve them. So that's a major medical need, which I think is very overlooked, actually. So when you look at the medical impact of cryobiology, I do think cryobiology is a small field compared to aging research. Obviously it's tiny compared to fields like cancer, but it could have a massive medical impact from organ banking for transplants, which I think is the obvious and major unmet medical need. If we could build organ banks for transplant, as I said, it would result in thousands of lives saved every year. But for other topics like reproductive medicine, like storage of stem cells and engineered tissues, you would have a lot of medical applications. And even more futuristic applications like in space travel. When you think about going to a different star system and taking decades of travel, that would require, at least one option would be to have some sort of medical biostasis or some sort of cold biostasis. And of course, more advanced also in terms of human medical biostasis. If you have a terminal condition, you'd be cryopreserved instead of dying. You'd be cryopreserved in hope of one day having a cure for that disease. Now, on this topic, actually, I wanted to point something else, given that this is a conference on drug discovery and aging, which is that, for the more transhumanists, and we had a talk today already on mind uploading. The point is that if you're trying to cure aging with drugs, I don't think that's possible. This is a slide that's not online yet. It's not been published from our drug age database of longevity drugs. And what this shows is that the longevity effects of different compounds, basically specifically from the ITP, the Interventions Testing Program of the NIA/NIH. So what this shows is really that the lifespan effects of these drugs is relatively small. The maximum increase in average mean lifespan is 26%. The maximum increase in maximum lifespan is 16%. And that's both from rapamycin, by the way. So we know rapamycin, I think, very promising compound, but it's still relatively limited lifespan effects or lifespan benefits. And that's the best performing one that we know so far. So the point is that the longevity drugs that we are identifying now, although they're promising, I am very optimistic about translating longevity drugs to the clinic. For more transhumanist purposes, we're not going to cure aging with these longevity drugs. In fact, their benefits are relatively small, and this is in mice. We don't even know how much benefits they would have in humans. So the alternative would be something like, as I said, medical biostasis, where you put life on hold. And this is a field where there has been some advances. This was the cover of Science magazine last year. This was a technology from colleagues at the University of Minnesota, I think, on developing a nano-warming system. So essentially these iron nanoparticles. So one of the problems of cryobiology as well is not just the cooling down, but it's the rewarming afterwards. So basically what they have is they have these iron nanoparticles. So they perfuse an organ with these iron nanoparticles before it's cryopreserved. And so then for when the rewarming, you use a magnetic field, and that leads to a homogeneous rewarming of the organ. Otherwise, the outside would rewarm faster than the inside, which can lead to all sorts of problems. So it's quite a very interesting technology, which I think they successfully applied to, if I remember correctly, rat kidneys, which they could cryopreserve, rewarm, and then transplant back to rats when the kidneys, the cryopreserved kidneys, were functional. So of course, rat kidneys are quite small compared to human kidneys, so there's still issues and there's still challenges to overcome, but it's a step in the right direction. As I'm sure you're aware, there's a lot of inspiration we can take from nature. Just like in longevity, we can take inspiration from animals like naked mole rats and bats and bowhead whales. In cryopreservation, we can take inspiration from nature, from some animals. The classical example is this you see here, the wood frog, which partly freezes during winter. So these and other animals, they secrete/produce different types of antifreeze molecules and proteins that we can try to understand and then apply that to human cryobiology. This is from, I think, from the lab of Ken Story in Canada, which is one of the leading experts on this topic. So in terms of what we are doing in this context, I mean, my take, I mean, a lot of my work has been on applying and developing computational methods and data-driven methods for different problems, longevity, aging, cancer, Alzheimer's disease, and so on. So trying to apply some of our data-driven methods and try to apply them in cryobiology. So one of the things we've done quite a bit in terms of longevity drugs is trying to apply different computational methods, machine learning, AI, deep learning methods to try to predict new longevity drugs. So that's something we've done quite a lot, and I've actually mentioned it in previous ARDD talks, and most of this has been published, but the idea is that we take all these large data sets and we develop a prioritization function for different longevity drugs, which we published over the years, including, well, in 2022 with Alexa Varankov and in silico, and with other labs as well. With Colin Hewald, who was right there more recently last year, and also Vadim Gladyshev on rilmenidine that we published last year. But the point is that we can take these large data sets of compounds, and we can look at the properties of those compounds that are associated with longevity, and then we try to identify other compounds with those same characteristics, which we can then test experimentally. So that's essentially the basic principle. And we apply that to longevity and aging. You can apply it, well, we've done a little bit on cancer as well, and you can apply it to many different diseases and many different processes. So the goal of what we are trying to do now is trying to do this for cryobiology. So we take molecular profiles of cryoprotectants, and then we develop different models and data analysis to then try to predict new cryoprotectants and new combinations of cryoprotectants, including using toxicity neutralizers, so other compounds that minimize the toxicity of the cryoprotectants as a way to improve cryopreservation. So that's something that's ongoing. So very briefly, last couple of minutes. So we started a new company in Oxford called Oxford Cryotechnology, whose goal is to harness these methods and technologies to develop new methods. So to use computational/machine learning data-driven tools to create new methods that allow us to develop new protocols for the cryopreservation of cells, tissues, organs, and ultimately people. And so I leave you with one thought, more futuristic, because one thing I'm convinced is that the cryobiology is an engineering problem. We understand the challenges, we just have to engineer how to overcome them. While aging is still a fundamental biology problem. We don't understand the drivers of aging, we don't understand, we can sometimes even agree how to define aging. We can sometimes not even agree how to spell aging. But in cryobiology, on the other hand, we have an understanding problems that have to engineer or develop ways of overcoming them. So, I mean, I'm convinced that we will have reversible human biostasis long before we cure aging or it's all of its diseases. And so I do think there will be a future where, a patient that has a terminal cancer or dementia or a terminal condition where they will be cryopreserved for how long it takes until a cure is developed. And that will completely change the medical establishment and healthcare. So I am convinced that's going to happen because I see that biology developments will happen way before cure all of these diseases of aging. And so with that, let me summarize. I hope I convince you that there is this huge need, a huge unmet need for better organ preservation protocols. I mean, there's a huge waiting list of patients that require new organ transplants that healthcare systems around the world cannot cope with. And so if we could safely and effectively cryopreserve human organs like kidneys, hearts, lungs, livers, etc., we could have organ banking, which would have huge medical benefits. It would be a huge longevity technology. And I hope one of the take-home messages of my talk today is that the cryobiology is a longevity technology with a huge potential for impact. In addition, there will be multiple other biomedical applications of cryobiology in different fields: regenerative medicine, reproductive medicine, and more futuristically, even space travel and medical biostasis. And then I've told you of what we're doing very briefly in terms of trying to apply machine learning, data-driven methods to improve cryopreservation protocols, because that's really one of the essential steps in allowing for organ banking and all of the applications. And so with this, I thank you very much for your time and attention. More information about our lab is available, and also about our work on cryobiology. Thank you very much.
Thank you very much thank you for introducing us to cryobiology. We have time for 2-3 questions.
Should I start?
Yeah, go ahead. Here. No, we have a microphone.
Yeah, I think that one of the fields that are maybe around the corner is taking ovarian tissue, freezing it, and prolonging menopause, knowing that the later the menopause, the longer the life of the woman. What do you think about that?
Absolutely. I completely agree. I think I briefly mentioned reproductive medicine, the areas of cryobiology, but that's perfect. Thank you very much. And I mean, I'll pay you later for asking that question. Okay, thank you.
We'll share the money that comes out.
Yeah, good.
Wayland?
Hey, over here, maybe?
Go first and then.
Thanks. Great talk. On the question of medical stasis, what do you think the ethics of that are? I think that's something that the field has to grapple with eventually, is this trade-off between people freezing themselves for future health discoveries and people participating now and actually being patients in trials to bring about future health discoveries.
That's a good question. I mean, when you say now, I mean, there are cryonics organizations that already provide cryopreservation services. I mean, my take on the subject, I should say I'm not signed up for cryonics myself. I think it's very unlikely it will work. Having said that, if someone tells me tomorrow I have a terminal cancer, I'm going to die in 2 years, I'm not sure how would I react. I think different people react to those kind of news in different ways. So I wouldn't necessarily, I don't even know how would I react. Who am I to judge people who sign up for it? And of course, in the end, from an ethical perspective, it's their body, it's their money. I think if it doesn't harm anyone else, why not? I mean, I have a very liberal view on that. If it's your body, do with it whatever you want. Okay, that opens a lot of can of worms as well, and I know. Okay, let's not. I'm talking on cryobiology, and let's not divert into different discussions.
One last. No, here, Wayland, go ahead. Yeah.
Thank you for the talk. It's very interesting, and I'm quite interested about the AI and machine learning model you are establishing, trying to find the new cryoprotected. So I would like to know what is the major outcome of this, because I can foresee there is a lot of aspects like the cellular function maintenance and the cellular structure and also the connective tissue, because we don't want to wake up with cancers, etc. So how would you like to balance everything into your model?
Okay, so that's a good question as well. I mean, what we're doing is a bit simpler than that. We're mostly focusing the model on compounds, chemicals. So you look at the properties of the chemicals, their chemical properties, their biological properties, that is, their targets, and you can use that information then to build a model of what constitutes an effective cryoprotectant, and then try to apply that to other chemicals or to toxicity neutralizers. So we're not modeling what happens in for example, during vitrification. We're more modeling, applying these models more for drug discovery and drug prediction. So that's more the context that we are applying this. I would say the rate limiting step is that there's not a lot of data on it. Because, for example, if you want to apply machine learning to cancer, it's relatively easy because there's so much cancer data out there. In cryobiology, in aging, we can do it to some degree as well, even though there's less data than for cancer. For cryobiology, there's even less data, and that starts to become a problem because, no matter how clever your AI is, if it doesn't have good data, it's going to struggle.
Thank you.
Thomas, very quick.
Very short. There are people that already hired those services where one can freeze themselves. Currently, these people that are on those services, what are the challenges that they are facing with? Because you mentioned that they might get crystallized or stuff like that. So, what are, for example, for them, what are they waiting for apart from curing whatever killed them?
Okay, that's a quick question, but it has a very long answer. So, because there's various challenges. First of all, is ice formation. Well, first of all, it depends on how a patient dies. I mean, if you die overnight from a stroke, and you can only be cryopreserved after 12 hours, having internal bleeding in your brain, obviously perfusing the brain with cryoprotectants becomes much more complicated, and you might have ice formation in the brain, which causes all sorts of problems. So that might be a problem. Even in an ideal scenario of cryopreservation of an organ, for example, you can still have some ice formation, depending on how it goes, basically. And then you have the issues of toxicity from the cryoprotectants. So you can have different sorts of problems that occur. And when you're imagining, I mean, I'm not aware of any study of like a patient that's been cryopreserved and revived, or even a pig, for example. I'm not aware of any of them, but I suspect there will be a lot of things wrong with it. A lot of damage, so I suspect there will be a lot of issues that would need to be overcome. That's my guess. And I think that's the guess of most neuroscientists as well.
Perfect. Thank you, Petra.
Thank you.